It's really nice to see you, Dr. Yadav. Can you hear me?
Yes, I can hear you. You
Great,
can?
great. If you don't mind, our sharing will begin in earnest. Now, I have introducing you to our Chinese audiences about your research and about your academic background. Now, I would like to introducing you in English again to our audiences, okay?
Absolutely fine.
Today, my guest is Professor Yadav from Columbia University. He leads System Biology of Aging Laboratory, which aims to identify novel functional connections between organs and to unravel factors that underlie human metabolism and its disorders. Welcome to our broadcasting room, Dr. Yadav. Our audiences are mainly anti-aging enthusiasts. They cannot wait to listen to your share on the fascinating discovery on the life-extending effect of taurine. Now, if you don't mind, shall we start?
Yes, sure. So as you all know, human society is aging. Aging is an inevitable process. All of us are going to age at one point in life. And this process of aging leads to a variety of changes in our bodies. They include different blood molecules, different cellular changes, which make us susceptible to diseases. In our quest to identify molecules that change with the age, we performed a metabolomic analysis in aged humans. And this analysis showed that a lot of molecules in the blood were changing. And one of the molecules that was most dominantly downregulated in aged humans was taurine. This was done almost 11 years ago now in my lab. And this made us interested in what taurine is and what does it do. Taurine is not a new molecule. It was identified in 1827 by two German scientists, Tiedemann and Gmelin. And since then, it was known to be associated with a variety of different processes. However, for almost 140 years, nobody cared about taurine because people didn't know what does it do precisely and what functions does it regulate in the body. In 1950s, pet foods were introduced in society. So people were rich at that point after World War II, and they needed some kind of a diet to be given to their pets. So the pet food industry provided these pellets, which can be given to these pets like cats and dogs at homes. And this led to precipitation of variety of diseases in pets. One of the most striking aspect of this entire episode was that cats started turning blind. So they were blind, they were hitting walls at homes, they were dropping glass vessels. So this was a very distressing sight to see for the pet owners. And they made large efforts and invested and to a lot of money in scientists to figure out why their cats were turning blind. This understanding of blindness in the cats led to the discovery in 1975 that the blindness in cats is caused by a specific nutrient deficiency in their diets. And this paper was published in 1975 in Science by Hayes and colleagues, which showed that blindness in cats is caused by one molecule deficiency in their diet, and that molecule was taurine. 15 years later, another study was published in Science in 1987, which showed that cardiac abnormalities or heart failure is also caused by taurine deficiency in the diet of cats. These two discoveries led to an increase in interest in the society and scientists regarding taurine, what taurine is and what does it do in the body. And since then, taurine has been shown to be associated with a variety of health parameters. However, it is not known whether taurine affects the aging process as we grow older. And our observation that taurine abundance was low in aged humans led us to investigate what taurine does during the aging process. What we showed is that taurine abundance declines with age in mice, in monkeys, and in humans. This raised a very important question, whether this abundance of taurine that declines with age, is it a driver of aging and actively participates in the aging process, or is it a passenger of aging that is just going along for a ride, is a collateral damage of the aging process? To investigate this, we then fed middle-aged mice, which are equivalent to 50-year-old human, 45 to 50-year-old humans, with once daily taurine supplementation for life. These animals that received taurine for life lived longer. On average, females lived 12% longer and males lived around 10% longer. Now, the next important question was, are these animals that are living longer, are they living healthier? Because you don't want to be living longer and being unhealthy for the entire period. That is a major goal of any anti-aging intervention, is to test whether animals that are now living longer, are they healthier or not? That is what we did next. Before we did, mouse is just one model organism. So we tested the effect of taurine supplementation in another animal model, in this case, C. elegans or worms. These worms also, which are multicellular non-vertebrate species, they also lived longer. On average, they lived up to 23% longer compared to control animals that received vehicle alone. But yeast, which is a single cellular species, did not have any effect of taurine supplementation. We identified that taurine mechanism of taurine and its synthesis was not evolved enough in a unicellular eukaryote. And as we evolved to multicellular species, to vertebrates, and to mammals, this taurine synthesis became specified and its metabolism got regulated and perhaps got selected to impart specific health benefits. Now, the next question was, are taurine-fed animals living healthy? And to test that, we next looked at a variety of organ system functions. We again fed middle-aged mice with taurine and looked at their health with variety of different assays for organ functions and terminal analysis of different parameters which cannot be assessed in live animals. This analysis showed that taurine supplementation had a small reduction in the body weight, and their fat pad weight, or the fat mass in them, was significantly reduced. This reduction in the adiposity or obesity in these animals was caused by a major increase in the energy expenditure. We identified that taurine supplemented animals were, in fact, they were eating same amount of food, but they were spending much more energy compared to their wild type or control animals which were receiving water. And this identified that taurine supplementation leads to increased energy expenditure, and that is one of the reasons why animals which were growing older, becoming obese in control situation, when they were supplemented with taurine, they had a reduced adiposity. When we looked at the bone structure now, the bone density in vertebra on the left-hand side was increased. The black lines here are bones. And you can see there are more black lines in the taurine supplemented animals. In a long bone, in the micro-CT images, you can see that the white lines, which are representing bone in the center, are increased in numbers. That showed that the bone structure was improved Not only bone structure was improved, when we looked at the bone strength by putting these animals through, meaning these bones through a biomechanical testing. So these bones were less fragile. They had more strength in them as well. So functionally, the bone function was improved because they had more strength. Next, we looked at, okay, how is the muscle parameter? So we looked at the grip strength of animals and we found that muscle strength was improved in taurine supplemented animals. You can see the 500 here is 500 milligram per kg body weight per day of taurine, and the 1000 is 1000 milligram per kg body weight per day. Compared to control animals, which is T0, you can see that the muscle strength was improved. Not only muscle strength was improved, their neuromuscular coordination in a rotarod test was improved significantly. This showed that taurine not only improves muscle strength, it also improves neuromuscular coordination, the ability of the muscular system to perform when put through different tests. When we looked at anxiety through a dark light assay, these animals which were taurine supplemented were less anxious, and they had improved memory. So in other words, taurine suppressed anxiety in animals and it increased the memory of these animals. Next, with age, we have a glucose intolerance, or we become type 2 diabetic. And when we looked at effect of taurine supplementation on glucose tolerance, you can see that these animals were able to clear glucose more efficiently compared to control animals. So their glucose tolerance improved. This improvement in glucose tolerance was due to increased insulin sensitivity in their tissues. They were more insulin sensitive as well. So these animals had an improved glucose and insulin tolerance. Now, with age, our ability to mount an immune response is ameliorated. It becomes compromised. And when we looked at the immunophenotype of these animals had a better immune system function compared to control animals. So, in other words, these animals were able to mount a better immune response, especially with a higher dose of taurine. Now, together, these studies tell us that taurine supplemented animals had an overall increase in the health span. They had a better muscle strength and neuromuscular coordination. They had better bone density. They had more pancreas functions. They had better gut motility, better gut motility. They had better brain function, less anxiety, more memory, better immune system function, less adiposity in their body, and they spend much more energy compared to control animals. At the same time, taurine supplementation did not affect the energy intake in the body. Their food intake was, on average, was normal compared to control animals. This tells us that the health span of these taurine supplemented animals was improved. So what I showed you so far is that taurine supplementation from middle-aged leads to increased lifespan in animals, and their health span was also improved, suggesting that taurine deficiency that happens with age is a driver of aging, because supplementing or reversing this deficiency leads to increased lifespan and health span. Now, the next question was, how does taurine suppress the aging phenotypes? And to do so, we now turned our attention to the way taurine acts in the cells. So taurine in the extracellular space or coming from the blood is taken up by a specific protein known as SLC6A6, which is a transporter that takes taurine inside the cells. To understand how taurine deficiency was regulating the variety of processes, we then knocked out in mouse taurine transporter. So in these taurine transporter deficient animals, cells are not able to take taurine in the cells. And this deficiency of taurine in the cells, the red line here is taurine deficient animals, which are induced taurine deficiency. These animals lived on average 50% less compared to control animals. So on average, wild type animals live, 50% of them are dead by 30 months of age. But in the case of taurine supplemented animals, around by 18 months, 50% of them are dead. Now, so taurine deficiency curtails healthy lifespan. It makes animals live shorter life. Now, how does taurine deficiency compromise cellular functions? To do so, we looked at a specific cell type here, which is bone cells, which make bone. And these bone cells express a lot of taurine transporter. And if you knock out taurine transporter, taurine goes down in these cells. These cells function less. They have a less ability to mineralize bone. And in vivo, they are able to function less. They have a less osteoblast numbers. They have less bone ability to form new bone. And when you supplement taurine in the control animals, the bone osteoblast numbers increase, osteoblast function increase. When you make animals deficient, these cells are not able to function in vivo as well, as can be seen on the right-hand side. So using these cells, next we performed a RNA sequencing analysis, which showed that taurine deficient cells had abrogation of variety of aging features. So with age, our cells get compromised. Our function of these cells get compromised. And these compromise in functions of cells can be described in terms of 9 hallmarks of aging. And these hallmarks of aging are telomere shortening, cell-cell communication get compromised, cells have more senescence, and there is an increased accumulation of these senescent cells, or zombie cells, we call it, in different tissues. They cannot sense nutrients very well. They have epigenetic changes in their DNA. They have more DNA damage and genomic instability. Their proteostasis goes down, their mitochondria are less functional, and they have more stem cell exhaustion. Overall, these changes lead to different aging features. And when we looked at the network analysis of this entire transcriptome, we identified that the senescence was especially enriched in taurine deficient cells. And this was forming a node in these genetic changes or the transcriptomic changes in the cells which are having taurine deficiency. So senescent cells with age accumulate in the body. These cells are also known as zombie cells, and they produce a lot of inflammatory cytokines such as interleukins, TNF alpha, and they lead to a variety of degenerative changes in different tissues. To understand whether senescent cells were contributing to this dysfunction, we looked at in vivo in taurine supplemented mice. The blue staining here is senescent cells, and in the brain, senescent cells are reduced. In liver, senescent cell reduced. In fat, senescent cell reduced, among other tissues that we tested. And when you have taurine deficiency in a taurine transporter knockout mice, you can see that the liver has more blue staining, fat as well as brain has more blue staining, showing that upon taurine supplementation, you have reduced senescence, and upon taurine deficiency, you have acceleration of senescence. To directly test whether senescent cells contribute to the dysfunction of taurine-dependent aging phenotypes, we now turned our attention to taurine-deficient mice, which have more senescence, and these senescent cells accumulate in their different organs. So we reason that if you remove senescent cells, these animals may live longer or not, and that is what we tested by using senolytic drugs, dasatinib and quercetin. So we fed taurine deficient mice with these two molecules that are known to suppress the amount of senescent cell accumulation. Their bone density improved. The senolytic treated animals have more bone density, more muscle strength, less anxiety, and their memory was improved. And these animals, which were given senolytics, lived around 20 to 22% longer compared to control animals, which are taurine deficient and were given water. So taurine suppresses senescence. The next parameter we looked at is telomere attrition. With age, the telomeres get accumulated, the telomere gets shortened, and now if you have telomerase deficiency, that means they have no telomeres. These telomerase deficiency animals have increased senescence in them. So you can see this is a telomerase deficient fishes here. And with the telomerase deficiency, there is more blue staining, that is increased accumulation of senescent. And when you treat them with taurine, the senescent numbers goes down. You can see the blue staining is reduced in 300 micromolar taurine and 10 millimolar of taurine. And this is quantified on the right-hand side. And these telomerase deficient fishes die, 40% or 50% of them die within 10 days after fertilization. And when you treat them with taurine, they have 100% rescue of telomerase deficiency induced consequences on organismal health. Next, we looked at DNA damage, and we saw that DNA damage was reduced by taurine supplementation. They had more, and in taurine deficiency, they had more DNA damage. And if you treat now with the DNA damaging agents such as paraquat, you have this red line here, which is paraquat treated animals. But when you treat them with taurine, they had improved survival in vivo in animals. So taurine suppresses DNA damage. So with age, we have epigenetic changes. And when we looked at epigenetic changes through methylation sequencing, we found that the taurine supplemented muscle from taurine supplemented animals and cerebral cortex from taurine supplemented animals was having a methylation pattern similar to younger animals compared to untreated animals, whereas liver was not changed. And their DNA, their histone methylation also was differentially regulated in different tissues. And when you have taurine deficiency, there's an increased methylation in taurine deficient animals, and their DNA age or methylation age in these muscles of taurine deficient animals was increased by almost threefold. Taurine impacts epigenetic changes in the genome. We looked at nutrient sensing and proteostasis. So one of the hallmarks of nutrient sensing and proteostasis is suppression. With age, there is a suppression of autophagy, and the protein translation get accelerated and regulating proteostasis. And this we showed that the protein translation was down in taurine supplemented animals, and their autophagy was increased. Whereas in taurine deficiency, we have reduced autophagy and increased protein ribosomal S6 phosphorylation. So taurine improves nutrient sensing in the cells. So to test whether nutrient sensing dysregulation was participating in taurine deficiency induced consequences on organismal health, we then treated animals with rapamycin that activates autophagy and increases protein synthesis. And we found that rapamycin treated taurine deficient animals had improved muscle strength, reduced anxiety, and the memory was improved, whereas bone density was not affected, demonstrating that several functions of taurine deficiency are mediated through at least autophagy or protein synthesis dysregulation contributes to this process. We looked at intracellular communication by looking at different inflammatory markers in the blood of these control animals and taurine supplemented animals compared to young animals. And what you can see here is the TNF alpha, interleukin 17 alpha, RANTES levels, and GM-CSF levels, and interleukin 1 alpha. The taurine supplemented animals was similar to the younger animals compared to non-taurine supplemented aged animals, showing that the inflammation that increases with age is many of the parameters of these inflammatory markers are suppressed by taurine supplementation in aged animals. We also looked at the stem cell parameters. Okay, what happens to the stem cell homeostasis upon taurine supplementation? We found that the skin stem cells were improved in numbers, And in taurine deficiency, you have a reduced number of skin stem cells. So taurine increases stem cell homeostasis, then improves the health of the stem cells among many organs that we tested. We next looked at mitochondrial dysfunction, and we found that mitochondria, with age, we have increased oxidative stress in the cells, which is caused by dysfunction of mitochondria. So the reactive oxygen species, which are damaging to our cells, accumulate with age. When we looked at taurine control animals, which are aged mice, which are placebo animals, and taurine treated animals, you can see that the mitochondrial oxidative species were reduced in taurine supplemented animals. Their protein oxidative damage was reduced. The lipid peroxidation, which is caused by oxidative species, was reduced. And not only that, taurine increased the mitochondrial biogenesis by looking at different markers here in the cells. In taurine deficient animals, these processes were accelerated. They had more oxidative stress. They had a reduced PGC1 alpha and UCP1 expression. So taurine improves mitochondrial health in animals. So overall, what we showed is that taurine supplementation leads to a variety of different changes in the cells. It suppresses DNA damage, it improves proteostasis, it increases autophagy, it suppresses senescence, it increases stem cell renewal in the tissues, and it improves nutrient sensing and intercellular communication. Now, these evidences told us that there is a improvement in variety of aging features that get compromised upon with age, and taurine supplementation is suppressing and improving these functions and making cells healthier. So these cells which are aged upon taurine supplementation are functioning better. So these changes in cells lead to better organ functions upon taurine supplementation and improves lifespan of animals. So taurine supplemented animals were living healthier and longer because taurine was improving variety of dysregulated aging features in the cells. Now, the most important thing one has to investigate is what happens in humans. So to address this question, we now looked at around 12,000 subjects, which are 60-year-olds, and measured taurine and its metabolite levels in the blood. For these 12,000 people, we also had all the health parameters available for them through longitudinal analysis. So we knew in these subjects whether they had more obesity or less obesity, whether they had more hypertension, less hypertension, more type 2 diabetes, less type 2 diabetes. They had what is their cholesterol levels? Do they have any problem in their platelets, blood cells, inflammation, liver damages? So when we perform this large analysis of taurine measurement and its metabolite measurements in these 12,000 people, we found that with age, the higher taurine levels in humans, in these 60-year-old humans, was associated with less obesity, they had less BMI, they had less diabetes, they had less random glucose, less liver damage, less inflammation, and among other variety of aging features. So what did this large data analysis told us is that higher taurine levels in humans, in aged humans, 60-year-old humans, is associated with better health in humans as well. So if taurine deficiency is associated with poor health in humans in aging, so we next tested what happens with different genetic markers. And we found that in a large analysis with the FinGen database in humans, that taurine mutations in taurine biosynthesis enzymes is associated with hypertension in humans. And taurine deficient humans have a poor eyesight or retinal degeneration. So if taurine deficiency in aged humans is associated with a variety of diseases, we next tested what about a healthy state of a human? So what is a healthy state of a human? Is exercise. So we next measured in exercising individuals, what happens to taurine and its metabolite levels. And we put sedentary subjects or couch potatoes, which don't exercise, natural bodybuilders, endurance athletes or marathon runners, and sprinters with a bout of exercise. And what we found is that taurine levels increased in these subjects. Hypotaurine levels, one of the precursor of taurine, increase in these subjects, and N-acetyl taurine also increased in these subjects upon exercise, suggesting that a bout of exercise increases taurine levels, and many of the health benefits of exercise might be mediated through an increase in taurine abundance in the blood of humans. So to check the biomedical relevance of these findings, we performed an intervention study in monkeys, because monkey is a very close species to humans. And we found that when you supplement monkeys once daily for 6 months and measure a variety of their health parameters, these monkeys had reduced body weight, they had less adiposity, they had better bone density, they had a better glycemic index or fasting glucose levels, they had less liver damage, they had better immune system function, and they had a less oxidative stress in their blood. So what it tells us is that taurine intervention in monkeys, which are closer to humans, also increases health of these animals. So overall, what I showed you today is that taurine deficiency is a driver of aging across variety of species, from worms, zebrafish, mouse, monkey, and humans. So the next step of this research, which we are currently envisaging, is performed a large multicentric multinational clinical trial to investigate whether taurine supplementation in humans also impart health benefits in a long term. And this is what we are currently putting it together for study design, because the goal here is now to investigate different ethnic groups, such as, for example, African Americans, white Americans, Chinese population, Indian population, Australian population, European population. How is this taurine level or intervention leading to changes in health of different ethnic groups? Because what we do not know at the present time, how is taurine metabolism changing in different ethnicities? Humans are outbred population. All of us came from African subcontinent long time ago. But we have evolved differently. We migrated to different regions of the world. And our dietary style changed. Our metabolism changed. Our environment changed. And it shaped these changes in the environment, dietary style, and our genetic architecture has shaped our physiology differently. So our goal now is to perform a clinical trial, multinational clinical trial across different continents to investigate the effect of taurine supplementation in the health of human being. And to do so, currently we are doing fundraising and finding clinical partners in different continents, because we want to investigate, does this taurine intervention in a large population, placebo-controlled population, leads to better health in humans? And we need to investigate in different ethnic groups. And as I said, we would like to investigate what happens in Chinese population, Asian, Indian, Japanese, Korean, American population upon taurine supplementation. And currently we are looking for clinical partners, including in China. We do not have a clinical partner in China as of now. So if any of the listeners are listening to us, and if they are interested in participating in these studies, not as a participant, as a subject, but a clinician who is ready to partner with us and investigate these health parameters upon taurine supplementation in Chinese population, we will welcome any contact from such clinical partners. And this study was done by across the globe in different countries, and we welcome any comments. And if you want to get in touch with us regarding clinical trial, you are happy to contact me or any of our colleagues across the world to figure out how you can participate in this clinical trial. Thank you very much for your attention. I am happy to take any question that you may have.
Thank you, Professor Yadav. Can you hear me now?
Yes, I can hear you.
Now, listening to your share, many of our followers have actually many questions to consult. May I convey their questions to you? Their enthusiasm far beyond our expectation.
Of course, I am happy to listen to any, and I will respond to it.
Um, Most of them are actually about the utilization or the supplement of taurine. First one is, what is the appropriate dosage of taurine supplements for us, for human?
So that is where I ended up my talk. If you now look at, I'll keep this slide, because what we are, I'm sorry, what we are in currently envisioning is a clinical trial. So before a clinical trial is completed, we cannot recommend any dosing. However, according to European Food Safety Authority, up to 6 grams of taurine is under the safe limit of human consumption per day. Now, we need, as I said very precisely, that we need to investigate how is taurine level changing in different ethnic groups. How is it changing in Chinese population with age? Is taurine causing a resistance in Asian population or not? We need to figure out what is the appropriate dosing regimen in a dose response clinical trial in Chinese, Indian, Korean, different populations to recommend any doses. And we are current, as I said, we are currently envisioning and putting together this clinical trial in order for us to take it forward. And once we have completed this clinical trial, which if we can raise enough funds, of course, in the next 4 to 5 years, we will have this information in different ethnic groups, including, I hope, in Chinese and Asian population, to recommend a appropriate dose depending on the age of the people that they have, and the level of taurine, the resistance of taurine, and what kind of deficiency is happening. And we are currently investigating all these questions to recommend a safe dose limit for different ethnic groups. And I am very particular here. We need to investigate in different ethnicities, what is the taurine metabolism all about? Before we do so, please bear with us. We have done this study for almost 12 years now. And now we need another 3 to 4 or 5 years if we raise enough funds and clinical partners in different countries to investigate it in humans.
Next question is that is it enough to just have food that are rich in taurine, or functional drinks with taurine is more suggested? And which one is better?
So when you look at the diet, although it has taurine, for example, the highest amount of taurine is present in shellfishes and these sepia and other species. It is deficient in vegetarian diets. It does come from milk to an extent. So even in the highest amount of taurine that is present in shellfishes, it is 1 milligram per gram of shellfish. That is, if you eat half kg of shellfish, per day, you get only 500 milligram. And the human effective dose is, according to the European Food Safety Authority, is up to 3 to 6 grams when you see the effect of taurine. So what it is not possible to obtain enough taurine especially in aged people with different disease conditions, to get enough taurine from the diet. And when it comes to now energy drinks, let me take you next question. Energy drinks are not the only taurine they have. They have high glucose, they have a caffeine. So one cannot take energy drinks and to supplement taurine, because these energy drinks for a long-term consumption, large volume consumption, has been shown to be associated with diseases in humans. So we only tested the effect of taurine here, not effect of combination of caffeine, glucose, and taurine. So we do not recommend consumption of energy drink to obtain taurine. And at the present time, we do not also recommend to go to the grocery store and buy taurine, because these taurine supplements in the market may be adulterated. They may have impurities in them. They may have other molecules in them. And we need to wait for the clinical trial to identify what is the specific effect of taurine in humans, in different ethnic population, before we recommend anything for the large human consumption. And we will have this information in the next 3 to 4 years.
And in your expectation, should we have taurine treatment for a long term or on a daily basis in the clinical trials?
In the clinical trial, what we are putting it together is going to be on a daily basis. We are doing a dose response study in humans with taking 3 or 6 grams per day. We are envisioning at the moment. And then we will look at different health parameters with different age groups, because we do not know the effect of taurine in different age groups in humans. So we'll have middle-aged humans, old age humans, and we will look at a dose response study to take this study forward.
And then up to now, on the animal experiments, is there any very severe potential adverse effects caused by taurine? No.
We have not noted any adverse effect in whatever function we tested. And if you just do a simple Google search, I have not come across a study where taurine supplementation has an adverse effect in health of an animal.
So can we say that it is a very safe one for animals?
It is a natural molecule. It is produced in our own bodies. It is produced from liver. And we can take it from the diet. It is not a synthetic drug. It is a natural molecule present in our body that is low in aged people. So now a natural molecule, it is like eating food. So now the goal here is to look at the safe limit of taurine in different age groups in humans, to identify what is a safe dose, because we need to, unless you investigate, you would not know whether any side effect exists, whether people with the kidney disease may have a problem with taurine supplementation, or liver disease may have a problem with taurine supplementation. We do not know, and this has to be investigated. Without investigation, one cannot comment on what can be the safe limit, because we need to look at as a whole human society. Humans are outbred population. We cannot say it works in European, okay, it works in Chinese population, no, or in Indian population, no. We need to investigate specific ethnicities before we recommend anything.
And some of our audiences have noticed that the taurine levels dropped by 80% in older animals, and can the supplementation of it restore their taurine levels to very youthful levels?
It does. That is what the dose regimen that we selected once per day leads to reversal of this deficiency.
I just found out that there is very professional questions. That did you use CSAD gene knockout mice to observe the lifespan changes and the effect after taurine treatment?
We did not use CSAD knockout mice, because in the absence of CSAD, you have other changes, compensatory changes that actually compensate for taurine production, because taurine can be still metabolized and synthesized. It can be obtained from the diet. It can also be obtained from the during the gestation by from the mother. It can also be obtained through variety of other means. So we did not investigate the effect of CSAD deficiency in the animal models. We only looked at taurine transporter deficiency because that is the best way to curtail taurine entry into the cells. Because when taurine transporter is absent on the cells, taurine cannot enter the cells, and it leads to a variety of changes and actually makes animals live unhealthy, and they die. 50% of them are dead within 15 months compared to 32 months.
I just come out with that there is one animal that in your research that their lives have not been extended by the taurine supplements.
That is the yeast. That is a single cellular species. So these are the very early eukaryote. During evolution, these cells came up together, the eukaryote. So prokaryotic cell and eukaryotic cell is a single cell. Yeast, which is actually made in baking, for example, the bread baking. So these yeast have not evolved the machinery of taurine synthesis and metabolism to utilize taurine for their function. And as we became from a single cell to multicellular species and to vertebrates and to mammals, we have now effectively utilized taurine or developed taurine biosynthetic and metabolism machinery to impart health benefits.
Then what does taurine say about the improvement of multiple aging markers in humans?
It tells us that if aged humans who have more taurine levels, they are more healthier because they have less obesity. These are 12,000 people. These are not small numbers. And if you look at these 12,000 people, higher taurine levels, people with higher taurine levels had less obesity, less BMI, they had less type 2 diabetes, they had less inflammation, they had less liver damage, and their immune system was better. So it tells you that taurine deficiency could also be a driver of aging in humans as well. But these are association analysis. And in humans, you can only do association analysis because nobody can measure lifespan of a human being. Because even if we start doing, no study has ever investigated that, because even if you do so, I will be dead by the time this intervention trial will be done in the lifespan of a human. But there are associations. If you look at the large, long-lived human population, like some Japanese islands and some of coastal regions of America or in Europe, these long-lived human population have higher taurine consumption in their diet. They have higher taurine levels in their blood, and they lived longer. So it tells you that there is association in humans, and we need to do the clinical trial to know whether taurine supplementation imparts health benefits in humans, and what is a safe limit of taurine in different ethnic groups to impart health benefits.
So we can conclude that it is a very promising anti-aging material, but also we need more clinical trials or even more animal experiments to see.
I think animal experiments I will be very confident about, because we have done very robustly across species. These results are coming from all over the globe, from different continents, Australia, Finland, Germany, France, Italy, Spain, North America, Canada. So we have this is done in across different, all the labs that have participated in the study come to the same conclusion. And that tells you that animal studies are robust. The only thing that we need now is a clinical trial in different ethnic groups. And as I said before, we are looking for clinical partners in China as well at the moment, because we need to have these clinicians who can help us investigate in a Chinese arm of clinical intervention to see how is the taurine levels in Chinese population, what will be the effect of taurine supplementation in Chinese population in a clinical trial? And I would welcome any interest from Chinese clinicians to take it forward. We already have partners in India, in Australia, in Singapore, in Finland, in Germany, in America, and I would like to see that a large Chinese hospital, a clinician comes forward and say, hey, look, we can do it in China as well.
And there are so many Chinese readers or anti-aging enthusiasts have noticed that these experiments, it has hit us a lot for a long time. So I hope that, or I believe that there will be many Chinese volunteers that wants to join in or cooperate in this. And we also, Timepie will also join in to popularize or to propaganda on these experiments. I mean, clinical trials for our Chinese readers to join it.
We're very happy. We would be very happy to welcome any contact, and I put my contact details, and they're encouraged to reach out to me, and then we can bring them on board of this clinical trial.
Thank you for your share and your answers to all the questions. Now, although not willing to, our interview comes to an end. It is really appreciated that you share with us so detailed and fruitful interpretation on the life-extending effects of taurine, and especially on its clinical trials promising future. I thought. Hope that we could get close contact and wish everything goes well with the clinical trials and you, Dr. Yadav.
Thank you very much for the time, and thank you for inviting me to be on your Zoom call. And well, let us hope for all of us that it goes well, because it's a natural molecule and it can be easily obtained in the supplement or through dietary intake or by exercising simply. If we have these information in next 3 to 4 years, we would have a better idea about how it is working, and we can take it forward from there.
Thank you so much again. Let's call it a day.
Thank you very much. It was a pleasure speaking with you.